Hey, everybody, welcome back. Let's get started for this next session on computational methods development. Our first speaker today is Amit Singer, who did his PhD in applied mathematics in Israel and is now a professor in the Department of Mathematics at Princeton University. Amit has spent his career developing algorithms for Cryo-EM structure determination, specifically focusing on understanding and parameterizing heterogeneity in Cryo-EM structures and ab initio structure determination. And today he's going to be telling us about his new developments in heterogeneity analysis. Thank you.
Okay, thank you very much for the introduction. And thank you very much for inviting me to this wonderful conference here. Everyone can hear me clearly? Okay, good. So I'll be talking today about RECOVAR. So I pronounce it as RECOVAR. You can pronounce it RECOVAR, however you wish. It's a computational method that we developed for heterogeneity analysis in Cryo-EM. By we, I mean, we did it together with my former postdoc, Mark Giles, who really did all the heavy lifting in this work. Mark is now an assistant professor in our math department. So, and you're very welcome after or even during my talk to look at the code. It's on GitHub, or if you wish to look at the PNAS paper that was recently published. So for this audience, I think I don't need to say much about heterogeneity analysis. People consider two types of heterogeneity in the data. One is discrete heterogeneity, when you have a few major classes, and traditionally, the computational method developed in the field was to do this type of 3D classification. Continuous heterogeneity, when you have some type of movement, flexible motion. And this is considered to be more difficult computationally to analyze. And there is like really a flurry of methods that have been developed in recent years to tackle this problem. And also this I consider perhaps like one of the major computational challenges in the field, and the journal like Nature Methods declared as like one of the methods to watch in 2022. The method developed really range from using all types of algorithms and methodologies. And nowadays we're like in the machine learning era, machine learning, deep learning, AI. And you can also see that in the methods for heterogeneity analysis. So methods like Cryo-Dragon, which is considered like one of the state-of-the-art methods that is based on deep neural nets, and another method that really leverage this idea in Dynamite, she also is here, that use deep neural nets. And we'll see today, maybe I'll try to convince you that there is still room for classical, traditional applied math and statistics for at least in this field of heterogeneity analysis for Cryo-EM. So all these methods basically operate in three stages. The first step is to embed the images, the Cryo-EM noisy images, in a common coordinate system that we call the latent space. So basically the Z coordinate, or the Z vector, if you wish, describe the conformational state of the molecule. That's what we wanted to describe. And somehow we want to do this mapping that takes images, not even volume, just very noisy images, and to decide what conformational space it corresponds to. And then in step two, to map those latent coordinates to 3D volumes, presumably at highest resolution possible. And then the third step is motion estimation. So say if you give me two latent coordinate vectors, and I want to find the path, the transition path from one state to the other, and show you this nice movie of how the molecule actually changes the structure. So for example, in Cryo-Dragon that I mentioned earlier, that developed by Ellen Jong, who is now a professor at Princeton, a colleague of mine. So step one, for example, it's a deep neural net. Step one, for example, would be the encoder part of the network. Step two for reconstructing the volume with the decoder. And there is not a lot of motion estimation in Cryo-Dragon. You can say, for example, find the shortest path in the graph. There are some issues with that, but But anyway, those are like this, let's say if you use ProdRug, and those would be like the 3 steps. In our work in RECOVAR, step 1 will be just PCA, but some variation of PCA because we cannot apply PCA directly to the data here. Step 2 would be kernel regression, and I'll go into the details of those 2 steps, obviously. And step 3 would be to estimate the density of the conformations and the free energy landscape, and based on which I'll show you how to find the motion. Okay, so we don't have a lot of time, so I'll not go into all the mathematical details in each of those 3 steps. I'll just give you maybe the big picture, and then you can go on and read more. So let's start with the first step of PCA, how to estimate the principal components. And typically, for those who know PCA, it starts with estimating the sample covariance of the data. So let's say that we have data points x1 to xn. Okay, so I have the xi's here, and you can estimate their sample mean, mu, and then form the sample covariance matrix, okay? We cannot really do this in heterogeneity analysis in Cryo because we don't see volumes. All we see are 2D noisy images. So that's already a challenge, okay? We only see projections, but so PI would be projection of xi to get the image plus noise. So we don't get to see volume. That's the first challenge. And to deal with this issue, so already like 10 years ago, I had a brilliant undergraduate student, Jim Katsevich, and his father. We worked together and we came up with a least squares approach to deal with problems that really leverages the Fourier projection size theorem. Then we have also an issue that, okay, how are we going to compute such a huge matrix? Okay, so in Cryo-EM, the volumes are of size like hundreds, about like, I don't know, 100, 200, 500 cubed. So the covariance matrix will be huge, okay? It will be larger than a million by million matrix, and you don't want really to do such large storage and to deal with such large storage and computation. And finally, you're also dealing with very noisy and limited data in a sense, again, you don't see the X size only projections. And also the data in heterogeneity is very, very noisy. So, so to deal with the computation issue, instead of actually forming the entire covariance matrix, we leverage on very recent advancements in randomized numerical linear algebra. So instead of computing the entire covariance matrix, we're just going to use a few columns of that covariance matrix. And we can actually do that, okay, using the least squares approach that we have. And we do it in the Fourier domain, not in the real domain. And one very important ingredient in this story that has to deal with the very noisy limited data is to use regularization. So here we really mimic more or less the same approach of rely on. So also in rely on, the RE stands for regularization. And so basically you can split your data into 2 independent subsets and compute the same columns of the covariance matrix from each of the 2 sets and compare them and compute what is called Fourier shell correlation. And from the Fourier shell correlation, deduce the signal to noise ratio and use it for regularization of the covariance. And that's really an important ingredient in RECOVAR, okay, or RECOVAR. Once you have the principal components, at least in principle, you can then expand each volume xi as a linear combination of the mean, say the homogeneous reconstruction, and a linear combination of the principal components with the z's being the latent coordinates. So now for every image, I can actually get at least an estimate of those of the latent coordinates and do step 1. Okay, so here's just an illustration of how principal components would look like for 3 different data sets from Empire. The runtime is not so bad. Okay, so if you work with something like 300,000 images, it will take like about 1 hour on 1 GPU. Okay, now let's move on to step 2, how to do the 3D reconstruction at high resolution, hopefully. And to make things simple, at least for the sake of exposition today, let's assume that I only have like only one latent coordinate z. Okay, so just one-dimensional conformation heterogeneity. So this would be described with this x-axis here. And if I just focus on, let's say, one voxel, or if you wish, just one Fourier component of the volume, as you move along the conformational space, okay, now it's just one-dimensional coordinate, you'll get some variation, okay? That's the conformational variability. So let's say that this is the true curve, which obviously we don't know. If we look at the images, okay, they are very noisy. And let's say that for each image I know or estimate what is the z coordinate, I'll get lots of noisy samples, okay, because the data is very, very noisy. So now the question is really how to estimate this true curve from the noisy samples that I have here, okay? So obviously what one would like to do is some kind of averaging, okay? So if you give me two noisy images, I get it. And now let's say if I have a new, say, stopped working. Okay, if I get a new point, how do I estimate the value here? So because we're dealing with noisy samples, the obvious thing to do is to do some kind of averaging. Okay, so there are two kinds of averaging that you'd like to do. One is to average all the values, say that as similar z value here, okay, because they are noisy, let's average them. But we also want to average, let's say, values of nearby z coordinates, because the function is smooth, okay, so it doesn't change so much. So let's also use other images, okay? Otherwise, we're going to end with maybe too few images. And then the question is how to choose this h, okay? How to choose what is called the kernel bandwidth, okay? So if you choose it to be, say, too large, okay, then you're going to average maybe too much and see, and you're going to do too much of smoothing, okay? If you're going to choose it too small, you're going to have too few samples, and then the noise will dominate. Okay, so there is some sweet spot of how to choose this h in an optimal manner, and what I just described, you can formulate it as in the form of kernel regression, okay? Where also the average weight, okay, will be, for example, proportional to how far you are in the z coordinates. So there are all kinds of kernels. For example, in reconstruction in realms, they use the triangular kernel. Here you can also use what is called the parabolic kernel, which is a bit more optimal. And you can see that if you choose the width to be too large, you get too much smoothing. If you choose to be too small, you get very noisy estimate. And there is a sweet spot, okay? Here it's the red curve, which gives you a nice smooth estimate. So indeed, you can do the analysis, okay? There is a famous bias variance decomposition of the mean squared error. You have two terms. One is the bias term that here will be proportional to h to the 4. There is the variance term that is proportional to 1 over the SNR. Obviously, we have to deal with the noise level here and 1 over h. And you can see now why there would be, you want to choose h to be too small and not too large. One thing to keep in mind is that in Cryo-EM, the SNR is also highly dependent or variable in the Fourier domain. So you're probably all familiar with the B factor, okay? So the SNR really deteriorates as you go to high frequencies, which means that the optimal h, okay, that we have to choose will also be a function of the frequency. So you don't want to choose like one h for all the frequencies in the data. And the same story also goes in real space, okay? So you can have type of movements, okay, that part of the molecule will not change as you have the motion, but other parts will move and will change considerably, okay? So this means that for the bias term, because there is no movement here, you can actually average all of the images in the data, okay? They all give you the same information that you want to average, but there are other parts of the molecules that you have to be very careful and only average a little bit, okay? Otherwise, you'll get a lot of smoothing that you don't want. So you want to do this this H, picking H in a smart way, and we do it in an adaptive way, both in the Fourier and real space. And now let me show you some results for reconstruction. So this for, again, this Empire data set of the ribosome. And I hope that you can see that for example, the Cryo-Dragon reconstruction has also sort of like noisy artifacts that you don't get them in the RECOVAR. And also like part of the ribosome is even like partly missing here. So this is like for one state. So here we like know the ground truth. So this is like a result of like 3D classification published about 10 years ago. Again, you can see this is like for a different state in the data. And you should be able to see like finer details in resolution details in the recovery construction. And I can also zoom in a little bit. So here you see, I don't know, in the zooming places where you get like finer details, like also for the red part, okay? At least like you can get the idea why, at least like, okay, like visually you get like high resolution. So it would be nice if there would be like, I don't know, some independent benchmarking that would compare different methods in for heterogeneity analysis. And luckily there was one very recently. So this is the work of, again, of Ellen Jog and her student Minkyu and collaborators. And they compared a few methods. Sure, so I don't think that dynamite is here, it was too new. The numbers here correspond to, it's a measure of resolution. It's basically like the area under the FSC curve. Okay, so the larger the number, the better. And usually those papers, like the bold number is like the highest one, and then the underline is the second best. And so two things to see here that for most of the simulated data sets here, synthetic data sets here, RECOVAR get the highest resolution. And moreover, if you look at the embedding itself, okay, so for example, for the first one, it's just a 1D type of motion. So it should be like just a closed curve. You see like really nicely how RECOVAR gives this curve. The other methods somehow like thicken that curve, and you may even confuse to think it's like a two-dimensional type of motion, and there are some other artifacts here that are not really in the data. So yeah, so this will go you through a few of the data sets. I'm not sure if you want to see all of them. About the running time, you can get the impression that I don't know, Cryo-Dragon is much faster, but they only reported the running time for one epoch of the neural network training, and so really you need to like multiply this by 100 if you want to do all the iterations. For RECOVAR, it's the entire time that to estimate the covariance matrix. Okay, so let's keep this for now. Okay, and let's go to the third step, the last step of estimating the motion and conformational landscape. So really what we are hoping for is something like this, to get like the energy landscape, to see where the potential wells, and given two states, I want you to be able to tell me what will be the transition path from one conformation to the other. So for this, I need to estimate somehow the density. Okay, from the very noisy images, I need to estimate the density in this latent space, because we know that Boltzmann statistics tell that the free energy is just a function of that density. Okay, so that would be really nice, but in reality, we get something like this. Okay, so the data is really, really noisy, and if you look at the embeddings, doesn't matter if it's like RECOVAR or 3D Flex or Cryo-Dragon, they all look more or less like blobs. And so it will be, I don't know, a bit for wishful thinking to say, okay, I can get free energy landscape from this and give you like really good transition path. But luckily, at least for RECOVAR, we can do something more, okay? Because it is based on just linear projection, just principal component analysis, where the other methods use some I don't know, some deep neural net for which it's very difficult to say or to argue that we can actually recover densities. So let's start with some, I don't know, synthetic data set that I want to show you also through this data set, like one of the pitfalls that you should really worry about and be mindful of when you analyze conformational heterogeneity in Cryo-EM. So these like more or less like realistic data set. We have like 300,000 images, reasonable size, the SNR, pole distribution, constant variation. They're all, we chose to be more or less quite realistic. The details are not so important, okay? Again, if you just take this synthetic data set and find the latent space coordinates, it gives you those blocks, okay? And you can even play this game with just no conformational heterogeneity, just homogeneous data sets, okay? So, but I increased the noise by a factor of 3, okay? And again, it looks like blobs, and these blobs are just because of the noise. Now, what you would usually do in those methods, okay, so you start changing the, let's say, one of the latent space coordinates, let's say from left and right, to see like what type of heterogeneity you have in the data, okay? Here there is no heterogeneity, okay? I just took a homogeneous data set, and this is what you get, okay? So all of these methods, to some extent, even though the data was completely homogeneous, okay, because of noise, they will hallucinate heterogeneity, okay? So you should really be careful when you do heterogeneity analysis. And because you can imagine that you are seeing something that is not really there. Okay, so now let's go back to RECOVAR and see if we can actually fix this issue and get transition paths more reliably. Okay, so we want to estimate the density, okay, this blue curve here. And we get noisy images. And as I said before, every noisy image will get us an estimate of a latent coordinate. And there will be a mismatch because of the noise. So what does this mismatch mean? It means that the true density that we have, we cannot really get it from the data. What we really get is that density convolved with a Gaussian kernel, where the Gaussian is due to the noise, okay? And this is like unique for RECOVAR because it's a linear projection. In the other methods that use nonlinear embeddings, you will not get this nice convolution with the Gaussian anymore. So what you want to do at this point is to do deconvolution, okay? To deconvolve the effect of the noise in the data and somehow estimate a density that hopefully will be very close to the true density. Okay, so that's what we do. So remember that before we got some very crude looking blobs, and now we do the deconvolution, and we get a much nicer density estimate. And for that very nice density estimate, now we can actually find trajectories. So the way we find trajectories, we basically look for the shortest path that will take you from one say initial state to an end state. But the way that we do it is we don't just look for a short path, because this will just give you a shortcut in the data, okay? That's what you really want. So we penalize, okay, in this shortest path integral formula by 1 over the density. So if you try to go through a region with very low density, you get very large penalization. And to solve this optimization problem, actually it boils down to solving a PDE known as the kernal equation. And so we can solve it in dimensions, say 1, 2, 3, 4, even like 5. And basically this will give us the trajectory. And you see that at least for the simulative data, we get trajectories that mimic very nicely the true ones. And so here, even though originally everything looked like very blobby, after this deconvolution step and solving the kernal equation, we can find the transition path, estimate densities relatively accurately, and even give you the motion. So that was for a synthetic data set. I can also demonstrate this for experimental data sets. Again, so this is now experimental data sets, images, we have the embedding, kernel density estimation will give you this type of blobby thing, deconvolution. You can even now see a metastable state. We find the transition path, and you can get the motion. Okay, this is another experimental data set. Now you see three metastable states, and you can find motion in this data. And yeah, so at least we're very happy at that point. We also work with, I'm not an experimentalist, but we sometimes get like, we collaborate with other experimental groups that want to analyze conformation heterogeneity in the data. So there's like work with a group in Chicago. I'm not going into the details. So let me summarize. So when it comes to resolution, apparently it seems that like averaging works better than deep learning, okay? And just simple averaging, what I showed you today. But maybe more profoundly, I would say that in this era of machine learning, that everyone just say, okay, why don't you just use deep neural net and solve your problem? Sometimes there is still an advantage to think very carefully about your problem and use the classical applied mathematics and statistics. And if you do it right, you may even get better results, okay? And for the experimentalists in the audience, just be cautious, okay? That heterogeneity analysis involves very, very noisy data. The embeddings can be very noisy, and you can easily hallucinate things in the noise, okay, that are not really there. So again, you can find our Python open source code here, and I would like to thank you for listening, to thank, of course, my collaborator, Mark, who, again, did all this difficult work, and to thank the funding agencies that supported this work. Thank you very much.
Okay, thank you very much, Amit, for a fantastic talk. I just want to remind people online that we'll take questions on Slack as well. Is there anything in the audience while I pull that up? You have some micro?
Hello? Oh, yeah, thanks. Thanks, that was such a beautiful talk. So I'm wondering, in these trajectories, we kind of know a lot about these objects, right? They're real chemical objects, and we know a lot about bond lengths and bond angles. So every snapshot needs to like follow these rules, right? So what would be the best way of injecting that information, which would kind of help with the noise, I assume?
Yeah, that's an excellent question. So far, what I described does not use any such prior knowledge, so prior information. Definitely, if you have that prior knowledge, you can leverage it. Like one thing that one approach that I think would be really interesting is to combine what I described here, which is really like extracting information just from the noisy images, from the data, with something like, let's say, molecular dynamic simulations, where you are going to leverage all the forces and interaction between the atoms in the molecule. And I think that can be also like a very powerful approach, but you're completely right. You want to leverage that extra information.
Question.
We didn't have too many beers last night because there was a lot of stairs coming down. Thank you very much for the talk. That was really cool to see behind the hood. In that complete homogeneous data set where you saw this sort of Einstein flexibility from noise, did you find any sort of artifact that gave you a clue that that was in fact happening from the data set? I mean, it was happening, but if I wanted to have a control, I'm looking at my reconstruction and I see this flexibility. Did you find some kind of artifact that would let me know that maybe this was noise more than real, if that makes sense?
I know, that's probably like $1 million question, but maybe you can go back to the previous question. If that's not consistent with actually the bond lengths and the other forces in the data, that will give you a clue that something is wrong here. Okay, yes. I would say.
Okay, I was hoping for something like the phase randomized curve that you get from your reconstruction, but for flexibility of some kind. But I think you're right. Just looking at the biology of it would answer that. Well, thank you.
Any other questions? Shores? Yeah.
I mean, this is great stuff, and thank you for making the code openly available on GitHub. Perhaps now that you're here at the CCP-EM conference, you could also have a chat with Matt and Colin and so on, because having a Doppio type of plugin to this will further, will make it much easier for you to reach a large community of users, I think, and your software would definitely be very useful for them.
Yeah, thank you for the suggestion. Yeah, I'll happy to chat.
Thank you. There was a question over there. Yeah.
Thank you for the talk. I had a question about the interpretation of the latent space, both in your software and more generally, because you mentioned how mapping it to free energy landscapes, and to what extent is there a one-to-one correspondence between latent spaces and the underlying free energy landscape, or is that an assumption that you have to put in?
Yeah, so we assume equilibrium, okay? So you know that the density will be like e to the minus k Boltzmann t, okay? So we have a way to relate the density with the free energy in that sense, okay? You just take the log, and yeah, but like I said, you have to be very mindful of the fact that the density estimate can be very, very noisy, okay? So this means that the energy estimate can be also noisy, and it is especially difficult to estimate at regions where the density is very low, okay? Where you have very few images. And so the estimate, for example, of what is like the energy barrier might be very difficult, okay? Because you just have too few images there, okay, at the barrier. So when I say, okay, estimate the energy landscape, it should all be taken with a grain of salt, okay? It's only what we can do from the data.
I guess further from the question about maybe model, like geometric constraints and model building, and how only really for the states where you can.
Right, so I think that we're probably at the cusp of where to take it further, you have to do things like was suggested here, okay? So like to combine what you get from the data with additional information about like the chemical bonds and lengths and things like that.
Thank you.
Okay,
Okay, maybe last question from Maya. Yeah.
Hi, thanks for the talk here.
Okay.
I'm just wondering what happens if you have a strong preferred orientation, if you tested it on such data.
If
you have strong preferred orientation in your data.
Yeah, we haven't tested the case of strong preferred orientation. We should probably do it, but yeah, you can expect that everything will be more difficult in that case because no, I'm just being very honest, okay? It's
Sorry, I was just wondering, maybe you could try to correct for it and then reanalyze
so the issue, for example, is even like the covariance estimation, okay? If you have preferred orientation and you don't get like full coverage of all the frequencies in the Fourier domain, the estimation of the covariance matrix is going to be very difficult. The eigenvalue estimation is going to be very difficult. So probably the solution in that case, you just need more data. So if for typical heterogeneity analysis, they require, let's say, 100,000, 300,000 images, maybe you live in like million particles or 3 million particles, just to compensate for the fact that you have very preferred orientation in the data.
Okay, thank you. I think we have to move on, but thank you again very much, Amit.
Thank you very much.
Bye-bye.